Hello children, welcome back to biology class. Yesterday you have learned about the first topic in biology, that's a cell. And today we will start with the second chapter in biology, the structure of chromosomes, the cell division and a cell cycle. Yes, yesterday I told you about the cell, the complete structure of a cell and some of the cell organelles also. And today, so before the starting, the structure of the chromosomes, there is a small topic called as cell division. Yes, have a look at the cell division. What is cell division? Is that really essential and necessary for the life? Yes, of course, cell division is the most important in all the living organisms. The cell division, it is a fundamental characteristics. Cell division is the fundamental characteristics of life. Fundamental characteristics of a life. That is the reason why cell division, which is very important for all the living organisms. Have you understood? Yes. Of course, there are some important the structure and also the importance of cell divisions in all the living organisms. When it comes to the chromosomes, yesterday you have learned the structure of a cell. In that structure of a cell, there are mainly three important parts of it. The outermost layer called as a cell membrane, the just below that the living part of the cell called as a cytoplasm, and after the immediate cytoplasm, there is a one more important the part of the cell. It's a nucleus. Yes, inside the nucleus. Inside this nucleus, there is a small, the thread-like structures as I told you before. Yesterday classes. The chromosomes. Yes, the chromosomes are the most important things in our life. Why? The chromosomes. What are chromosomes? The chromosomes which are present inside the nucleus in the form of chromatin fibers, not directly in the form of chromosomes. After the cell division only, the sum of the chromatin fibers they converted into the chromosomes. See, what is the chromosomes? The chromosome says the nucleus contains the most important cells of a DNA, which is organized into discrete units, which is nothing but the nucleus which is contains, as I told you, the chromatin fibers. The units are called as chromosomes. These chromosomes are the darkly thread-like structures. Have you understood? And each chromosome contains one long DNA molecule associated with the many proteins. The complex DNA and the proteins, the formation are called as chromatin. The chromatin fibers, I told you, right? This is the most important. The DNA and the proteins, they are the most important factors in the chromosomes. Clear now? And the chromosomes, who discovered the chromosomes? The chromosomes was discovered by the famous German scientist called as the Walter Flemming. The scientist Walter Flemming, he was a German scientist. In the year of 1882, he discovered the chromosomes in the larva of the salamander. The salamander you guys knows very well, right? It's like an example for an amphibians, the salamander. And the thread-like, when he noticed with the handmade microscopes, inside that, the thread-like that appears in the dividing the lengthwise, he called as the lengthwise structures, he called as it's a mitosis. The division, as I told you before, the cell division. Yes, cell division, which is nothing but the division of a cell to form the new kinds of cells. It's a very important for the continuation of a generation. In the same way here also. So that's why the thread-like structures appears in the salamander larvae. We call it as mitosis. This is a stage of a cell division. Understood? In the same way, the chromatin. The chromatin formed of two substances mainly. The chromatin, it's a form of, it's composed of two substances mainly. As I told you before, it's a combination of the DNA and the proteins. Yes, the DNA. The most important characteristics of the DNA, It's all about the 40% of the DNA, and the proteins are nothing but histones. The histones are the proteins. They are all about the 60%. About 60%. The 60% are made up of the chromatins. But when it comes to the structure and molecules of the DNA, have you understood this one? The cell division, it's the most important characteristic feature of all the living organisms. The next one is the chromosome. The chromosomes, they are not directly in the form of a chromosome. Before that, the chromosomes, they will be in the form of the chromatin fibers which presents inside the nucleus. The later, the chromatin threads. That chromatin threads are chromatin fibers. They are composed of two main components. Such components, the DNA and the histones. Let us start with the structure of a DNA molecule here. What is DNA? The DNA is nothing but deoxyribonucleic acid. DNA, deoxyribonucleic acid. Nucleic acids. This is the full form of a DNA. Basically, the DNA, what is the function of a DNA? As I told you before in the yesterday classes, the DNA is a functional segment of DNA that's called as a genes. That genes which carries the characteristics from the parents to the offspring. The same way here also, the continuation of a generation, they should carry some of the characteristics from the parents to the offspring. Have you understood? Here, the DNA, when it comes to the structure of a DNA, the DNAs, they are the large, the macromolecules because of the single stranded, the long chain composed of two complementary strands. That's called as double helix model. For the first time, DNA double helix model was discovered by the two scientists, Watson and Crick. Watson and Crick. These are the two scientists who discovered the double helix model of a DNA. Clear? And the single stranded DNA is composed of the repeated nucleotides. On the surface of a DNA, there will be number of repeated nucleotides. Such nucleotides are called as the repeated nucleotides. Clear now? That nucleotides are made up of three different components called as the first group of components, phosphate. The second one, the sugars. The third important, the nitrogenous bases. Have a look here. These are the structure of the important, the components of nucleotides. The first, you can see this one, the circle shaped. This is the structure of the phosphate. Have a look here. This is the phosphate. The phosphates are always linked together. Which one? The phosphate, sugars and the nitrogenous bases. Here, the phosphates and the sugars are pentose. Pentose. The shape of the sugars are the pentose. It means the 5 orders will be there. The next one, the new nitrogenous bases. They are always bonding together to form the nucleotides. Clear now? I told you the single stranded DNA are called as the macromolecules. When the formation of the double helix model, the nitrogenous bases, they forms the ladder-like structures. Have a look at this diagram. See how it's look like exactly the ladder-like structures, right? This is a double helix model of DNA structure. This double helix model of a DNA, which was discovered by the two scientists, Watson and Crick. Have you understood? Here, the single-stranded DNA, they composed of the repeated nucleosides, the repeated number of nucleosides. The nucleotides especially, they are made up of three different types of components. Which are those? The first set, the first group, the phosphate. The second, sugars. The third, the nitrogenous bases. Especially when it comes to the 4 types of nitrogenous bases are there. Which one? The bases. Nitrogenous bases, mainly the 4 types are there. Which are those? The first one is the adenine, guanine, cytosine, thymine. These all the nitrogenous nitrogenous bases, they will be arranged on the surface of a nucleosides. Here you can see this is a double helix structure of a DNA. Now one ladder, it is a complete ladder. The single stranded DNA, the nitrogenous bases will be arranged. The opposite of one more ladder side, there will be nucleosides. How they are arranged? Always adenine paired with the thymine with the 2 hydrogen bonds, and the guanine always paired with the cytosine with the 3 hydrogenous bonds. Clear now? Yes. When it comes to, have a look at the structure of this one. Have a look, how it is forms from the single cell to the nucleus, the chromosomes and all the structure. Let us start. In a human cells, it's about the 6 micrometer inside. So in order to fix inside the nucleus, it has to be condensed and a coil and supercoiled. Clear now? See, because of that, we have a look here. The complete structure of a chromosome, it will be, look here. The rod-like structure, it's like an into mark. The sister chromatid, 2 hands-like structure. These 2 structures are called as sister chromatids. Sister chromatids. And between 2 sister chromatids, there will be the special structure are called as centromere. Centromere. On the surface of a centromere, there will be protection layer called as kinetochore. There will be the core which is formed on the surface of a centromere, that called as kinetochore. 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 T-O-R-E. Kinetochore. See, have a look at this one, the complete structure. The chromosomes, the complete structure of a chromosome, when it becomes a supercoil, like in a telephone. You have seen that the telephones, how the coil, it's like, the condensed RNA coil is supercoiled in all the ways. So this is called as the supercoils. And have a look at this one. This is a coil, just single layer, that's called as a coil. And the nucleosomes, what are nucleosomes? As I told you before, that's a nucleosomes. Here, on the surface of the nucleosomes, there will be 8 core histones to form the nucleosomes. There will be 8 different types of the histones will be there to form the nucleosomes. The DNA will be covered on the histones. When uncoiling of the DNA, there will be the formation of a new strand. That new strand which helps the formation of the double helix model of a DNA, the complete structure of a DNA. In each single strand, the arrangements of nitrogenous bases, always adenine, the base with the thymine, with the 2 hydrogenous bonds, the guanine always pairs with the cytosine with the 3 hydrogenous bonds. The same way here also, A, T, G, C, here vice versa. C always pairs with guanine. G always pairs with cytosine. So likewise. Here now, and when it comes to this is the complete structure of a DNA, double helix model of a DNA. Then, as I told you before, the 8 histone proteins, they are externally called by the DNAs. Then the nucleosomes. How many? The 8 core histoproteins to form the nucleosomes here. The coiling and the supercoiling of the DNA chromatin fibers inside the nucleus. When it comes to this is the nucleus. This is the nucleus structure. Inside the nucleus you can see, as we will study about the stages and the cell division stages in the next classes in the cell division topics. If you have a look here, this is the nucleus. Inside the nucleus there will be n number of the different sets of the chromosomes are there. Clear now? The next topic, the important topic is the cell division. In the first phase of an interphase, that is G1 phase. See, what are the things, what are the cyclic process which takes place in a G1 phase? That's the first important thing is the synthesis of RNAs and the proteins, and also increases the production and synthesis of and increases the amount of cytoplasm in a G1 phase. That's the most important thing which takes place in the G1 phase. And the next one, in a G1 phase, some of the different organisms like in plants and also animals. In animals, the mitochondria, in plants, the chloroplast, they will be having their own DNA, so that they can synthesize their own DNA. And here, there is no donation of the DNA from the other organisms. All the cells, whatever the cells, they enter into the G1 phase, they will be having either two chances. All the cells must follow the two paths. The two paths. Either they may withdraw the cell cycle. If there are any the weakened cells means the low strength cells. If they can't participate in the cell cycle, they can withdraw the cell cycle phase. If they are strong enough to entering for the next stage, that is the AS synthesis stage, they have the entering into the resting phase or the next cell division phase, that is in a synthesis phase. So this is the most important the points which has to follow in a G1 phase. Clear now? And the S phase, what is the importance of, what are the activities which takes place in a S phase? S phase is nothing but the synthesis phase. Have a look here. The synthesis phase, more amount of a DNA is synthesized and the chromosomes are duplicate. Yes, as I told you before, the more amount of DNA is very essential for the growth and development of an organisms, and the continuation of a generation also DNA has to be duplicate itself. Yes or no? All these, the more amount of DNA and also the chromosomes are duplicated when it comes to the synthesis phase. So this is the most important phase of in the interphase stage. And the third one, that's a G2 phase. That's a growth phase 2. That's the third phase of the interphase. The more RNA and the proteins necessary for the cell division, the synthesis of RNAs and the proteins, which will be double when it reaches to the G2 phase. That's in a growth phase 2. Because of when the cells are entering into the mitosis, that is in a stage 2, it has to be increases their amount of proteins and also the RNAs. It's very essential for the cell division stages. The cell division, it has to follow the certain stages. So that is the reason why they require more amount of RNA and also the proteins also. Have understood here? And after the G2 phase, they will be followed by the next stage. We call it as M phase or a mitosis phase or mitosis. Here, the mitosis, they will be following certain the different stages. There will be the mainly 4 stages are there. The first stages are prophase, the second stage is metaphase, third stage is telophase, the fourth stage is anaphase. These are the certain important stages of in a mitosis. And there will be the 2 most important stages in mitosis, that is special characteristics, the karyokinesis and also the cytokinesis. Yes children, today we have completed the cell cycle stage, and also in that cell cycle stage we have completed the first phase called as an interphase, and further the next stage called as a mitosis stage or an M phase, that will be continuing in the next session. Until, please subscribe the Global International Online Classes. Thank you.